That's always like the weirdest part.
I don't know. I'm just curious if there was like you listen to the lectures, you...
Right. So for this class, what I like to do was I would listen to the lectures at least like sometime within that week, like after we took the class. So maybe like the Wednesday, I would listen to Monday's lecture, or like Monday listen to last Wednesday's lecture, like before class started. And I felt like that at least I could remember what we talked about last week. So coming in, I'd be like, oh, okay, we just finished this unit. Now we're moving on to this.
When you were listening to it, would you go along with the PowerPoint and make additional notes?
Yes. Yeah. So what you may or may not have seen already is on the website, on his justknoweverything.com website, there's clinical cases, which are also included on your exam, but there's also these like Word documents that kind of comprise like all of the knowledge that you may or may not need to know. And so those notes were like a student's notes that just like he or she just wrote everything that, like ever came up. And sometimes it's superfluous knowledge, like it's a little bit farther than the extent of what we need to know in this class. But other times it can be helpful as another resource.
So you're talking about the lecture notes?
Yes, so the actual like Word document notes.
Okay.
Yeah.
But the PowerPoint is mostly where the bulk of those questions are going to come from. So typically it's like a 25 question exam, and of those 25 questions, maybe 7 are clinical based, and then the rest are like the content. And so with clinical cases, like I recommend getting like a group together and you just kind of run through them together. Like you can read through these clinical cases, they're kind of dense, but if you break it down onto your cheat sheet, like usually they're very straightforward in terms of like, okay, like a 55-year-old male presents with nausea, vomiting, blah, blah, blah. They run XYZ lab tests, which shows this. And so that kind of knowledge is good. And so what I brought is what my cheat sheet looks like. Just gonna show the camera. But if you guys can look this over and circulate it around while we're talking. But I like to organize the cheat sheet in terms of like sections, and you don't want to leave the cheat sheet till like the weekend before, definitely. So while you're going through PowerPoints, star things that like you'll probably miss or get tripped up on or something like that. Usually what I like to do on the cheat sheet is have like hard numbers or equations that'll come up. Sometimes just quick reference guides. You don't want to rely on the cheat sheet, but it's good to check. And we'll talk more along the way. Like if I see a slide or something or a table that I think is worth putting on, like I would just tell you, and then, from there. This is a copy of my old exam. You guys can like flip through it, and I don't know. Right now it's probably still like way over, but you'll get there.
Yeah, I printed most of these out and started like making notes on the test, just to kind of reword the question, or especially like given the answers, what is another possible question. Right.
So once things get rolling, I will try to provide questions sometimes, or try to incorporate questions into like these lecture PowerPoints that I'll be running through. But other than that, you guys can always ask me questions, always interrupt me, send me emails, whatever makes you guys comfortable or anything like that. I'm here to help.
Did you make flashcards when you were going through it?
What I would do is I would run through lectures with like other students, and then I would just kind of reread them over and over, and we would kind of like quiz each other. A lot of the stuff in this class is either it's very like point blank, what is this? Or it's, if this happens, what happens next? So if you can get like a group together to meet outside of class, like that's always good to bounce ideas off of each other. Depending on what unit, it's always good to just have that like, you hear it from someone else saying it, you say it, out loud, and all those different kind of stimuli are very good for helping you learn it yourselves.
Cool. Thank you.
Yeah. So, all right, let's begin. So I'm going to start with the very first PowerPoint, the cells lecture. So basically with the cell, there's many different organelles, and they all have different functions. And the types of questions that you might see are like, which of the following has this function? Mm-hmm. So really quick, I'm just going to run through all of them. So the nucleus of the cell is where you store all of your DNA, all of the genetic material that you need. And in general, the central dogma is what we call it, is when you have DNA can code for RNA, and then RNA can be translated into protein. And protein is basically what makes up everything like in the body, and it's very important for structural and like receptors, enzymes. It's very important all around. The nucleolus is kind of like that core of your nucleus, and the nucleolus is where RNA is made. So you're building that material to further make things in the future. Ribosomes, they are composed of two subunits of rRNA, so ribosomal RNA, and that's the site of replication for proteins. So what that means is I have my RNA, and I'm going to translate it along the ribosomes to make proteins. And ribosomes are scattered all throughout the cytoplasm of the cell, and then they're also embedded along what's called the endoplasmic reticulum, the ER. And so the ER, there's two types. There's the smooth and the rough ER. The rough ER is rough because you've got like all these ribosomes embedded on it. So then the rough ER is where you make proteins that are later secreted or used for transport or something like that. Whereas your smooth ER, that's for metabolizing your molecules, and it's for detoxifying the cell. It's got a lot of different functions, but typically it's very different than the rough ER. So be sure to keep those two in mind. We also have what's called the mitochondria, and the mitochondria is important for creating ATP, which is like the smallest subunit of like making energy in the cell. We also have lysosomes, and lysosomes are for garbage disposal, and they kind of break up things in the cell that we might find are potentially harmful or are just kind of waste products that we need to get rid of. And then lastly, we have something called peroxisomes, and they break up free radicals, which are like H2O2, hydrogen peroxide, something like that, it's similar to a lysosome by like breaking down some sort of unnecessary or unwanted product, but slightly different function, like slightly different substrate that it's acting on. So in the next slide, we have this slide here, I guess, or sorry, this one here. This one, sorry, this one. For endocytosis. So endocytosis is basically when a cell is kind of near something that it may or may not want, and it either like it kind of engulfs that in. And so there's either phagocytosis when you're eating something large, like a large molecule, or penocytosis, you're like cell drinking like a very small particle. And then exocytosis is the opposite. You're like pushing something out towards the rest of the cell. It could be, some sort of secretory vesicle, or it could just be something like that you just didn't want anymore in the cell.
Sorry to interrupt you.
Yes.
I don't remember him saying in class the different types of endocytosis. Did you do that? And I just totally missed it.
He mentioned phagocytosis and penocytosis.
Yeah. So like a lot of the stuff in the first PowerPoint at least, it's not too detail oriented, or it's not like the nitty gritty. It's more like the bigger ideas and foundation. So this kind of stuff kind of might orient you for later on. So I wouldn't stress too much about like every little detail that I may be telling you right now. For example, the next slide I have here is let's see, is the carbohydrate synthesis, or there's the phospholipid bilayer. And so in the bilayer, what we have are membrane channel proteins, and we also have a bunch of different other things embedded in the phospholipid bilayer. So you've got these hydrophilic heads, so these little beads that are along the top of the bilayer, and so they're made up of like the phosphate group of a phospholipid, and a lipid is hydrophobic, so it's afraid of water, so it points inward, and it's a bilayer because we've got these two phosphate heads pointing like on top and bottom, and we've also got like the hydrophobic lipid regions in the middle, like pointing inward. And so typically what that will tell us is when we have a hydrophobic particle, so something nonpolar, something that isn't aqueous, or something that isn't charged, that hydrophobic thing can get through the cell pretty easily. But then if we have a charged particle, it has to go through, we've got different proteins, ion channels, sometimes there's gated channels, or leak channels. There's a bunch of different types of channels that are specific towards some type of ion or charged particle that is trying to get through from one side into the cell. In this slide here, we have the plasma membrane proteins. So there's a bunch of different things embedded in that plasma membrane. So you could have a transport channel, so it just moves from higher to low concentration, and that's called diffusion. Passive diffusion is when you don't need any sort of membrane or channel or anything like that, and it can just simply just go through. But there's also facilitated, when you have a membrane that or a channel that actually, like, something has to go through to get from one side to the other. Like glucose is a large molecule, but it needs to be facilitated to get through from one side to the other. It's not so simple. Whereas, so you can also have different kinds of receptors like enzymes or cell surface receptors. So if something binds, all of these things that are embedded in the plasma membrane, they're all very specific. So they have to have the exact orientation, the exact substrate that it's looking for to bind to it, so that whatever functions will happen. And these are all just different examples of different things in the cell.
So do you think that is something good for, the cheat sheet? At this point you said
I think at this point it's you'll know it by the time like we get to it. It's one of those things where once you get to like a process, you're gonna be like, oh, that's this kind of channel, and then it'll kind of ingrain in your head. What I consider on like the cheat sheet, I feel like its space is very limited and it's like very precious. So like this is size 4 font, and like what I did was I made like Microsoft Word, and I just made tables, and I connected all the tables and formatted all the boxes. And if you make text boxes, they can be like easy to move around, but then there's like extra space on the edges of them. It's kind of weird, but you play around with it and you eventually can like format it this way. Also, super recommend finding people with color printers. Yes, color printers are great.
Just to go back to the bilayers, so the non-polar,
Yes.
not charged,
Yes.
they easily pass through
Yes.
the cell, but then the charged ones need a channel.
Right.
Okay.
Right. So there's different types of signaling that you'll come across throughout this course, and so there's hormonal things get secreted directly into a bloodstream, and then they kind of travel to wherever they need to act.
Sorry, Victor,
Yes.
do you have the slide where it talks about tight junctions?
You just said that one. It was back. You went.
I probably do.
It's okay if you don't.
No, it was right before that when I saw it. Yeah.
Oh yeah, that
This
one.
one, got it. All right, so with this one, tight junctions, they're also called zonula occludens,
Yeah.
and so they occlude different things. So they are like a zipper of a Ziploc bag. They're watertight but not mechanically strong. So they're in like your intestinal lumen. They're kind of like holding everything together, and they're making sure that like stuff doesn't just leak out or whatever. Whereas your zonula adherens, and that's your belt desmosome, those are like the seams around your pants. They're strong, but water can get through. Okay, so it's not watertight, but it's mechanically very strong. Your spot desmosome, like it sounds, it's in one spot, very tight. Gap junctions are, they're important for like your cardiac muscle or different smooth muscle. It basically, it's easy, they're like holes between cells almost, where you can kind of like signal one cell and signal the next. I think his example was like hotel rooms with a shared door. So if something happens on one side, then it can, signal the other door. And then lastly, there's something called hemidesmosomes. So they're like half of a regular desmosome kind of. So they anchor that cell to the basement membrane of the cell. Hold on, I'm gonna take a call from the other tutor. Hey Ricky, for your patho tutoring, did you manage to connect to the projector? Yeah. Uh-huh. Uh-huh. Mm. Okay. Okay, where's the HDMI cord in the back of the table? I mean, in the back of the TV? Like, I see on the right side. Okay. Alright. Sorry, guys. Alright. I mean, I see the cord, but I don't see the actual, like, slot to actually plug it in. What do I do? Oh, there it is. That's interesting. Yeah, it did. Okay. Alright, thanks. Alright. Yeah? Anything else? Alright, thanks. Bye. So guys, sorry that we're orienting this way now. Alright. Alrighty. Getting this to work. Okay, so let's see, where is this slideshow? Okay, so I hope people can see what I'm doing. Let's see. Yeah. Okay, so did anyone have questions about this particular slide anymore with the different types of junctions? Okay. So, as I was saying before, going to the different hormonal, paracrine, autocrine, those different types of signals. So a paracrine signal is something local, whereas an autocrine signal is auto, so think self. Usually in biology, auto refers to self. So if I say I autophosphorylate, I phosphorylate myself, I auto-signal. Neurotransmitters, they're from nerve cells, and from nerve cells they reach, I think we come over this in a little bit, the neurotransmitter will act on a receptor, and depending on where that receptor is and what type of receptor, then it'll have a corresponding function. So in biology, a lot of the actual molecule that's being secreted, it could be the same, but its actual effect, like the final effect, depends on what the receptor is of that specific cell, like that final destination cell. And I'll give examples once we kind of get to them. And then a neurohormone is like similar in that a neuron secretes a hormone, so something into the bloodstream that travels further down and then affects a target cell. So this slide here is about different types of channels, and they're good to know in terms of mechanism for like later on if you ever see anything. So a gated ion channel, that's typically like, it will only allow the specific ion that it's like, so if I have a sodium gated ion channel, only sodium can get through, or potassium if it's a potassium ion channel. A G protein coupled receptor, what that is some sort of molecule will bind to the top, and it has different components, and it splits up, and it activates what's called a signaling transduction cascade. And so that cascade will affect hundreds of different molecules in the cell to do some sort of effect. And it's just another type of receptor that you'll come across. Receptor enzyme can just be another ligand, and a ligand is the thing that actually binds. It could be like that ligand binds to the enzyme, and then the enzyme does XYZ activity. A steroid receptor is a little bit different than the other receptors, because the actual receptor is along the nucleus, the nuclear envelope. Because remember, a steroid hydrophobic, a steroid is a type of lipid, hydrophobic, a fat. Those are all kind of semi-interchangeable. And so fats, lipids, hydrophobic, or steroids, they come in through the outer membrane of the cell, and they act on the nuclear envelope. And then they activate gene expression, or they modify the transcription or other things in the cell to make more protein, or make less protein depending on what kind of hormone it is. And then there's these other kinds that you don't really come across as much. So here's what I was saying earlier. So hydrophobic molecules, so things that are non-polar, they can get through just fine. Small, slightly charged, also just fine. Like water, just fine. Glucose and sucrose, however, they're pretty large, so like you typically need help. So because they're so big, they need a little bit of a push. And they're just a little bit charged, but not like particularly, so they're pretty much uncharged, just big. And then ions are like a no-no. They absolutely need a channel to get from one side to the other.
What channel would glucose use?
So there's a specific channel called like GLUT, GLUT, or like specific ones like that, and we'll come across that soon. So something like this, where it has like all these numbers, that's something that I would put on the cheat sheet, because sometimes he might ask you like, is this a normal value? Well, not specifically ask you, is this a normal value, but it's more like, the patient has this, like these numbers, what kind of clinical manifestation might he have? So always try to orient yourself. So like sodium, so there's a difference between being inside and outside of a cell. And so that's what this like ECF and ICF kind of part are. So sodium is more on the outside of the cell. And then potassium, that's the K here, that's more inside the cell. So I don't know, have you guys ever heard of like Special K, the cereal? So like Special K, like the K is special, it's on the inside, and then like everything else is like all on the outside is more. I don't know if that can help, but like that's how I think of it. Special K, more on the inside. Most everything else is like greater. So like sodium is greater on the outside, chlorine is greater on the outside because you got a greater sodium on the outside to kind of balance that electric charge. Same with calcium, it's also bigger, and like bicarbonate, this is HCO3 minus, that's important to buffer the cell against hydrogen ions. Hydrogen ions are H+, that's kind of with this number. So are you guys familiar with like the pH scale? So when you have more hydrogen ions, more H+, that means your pH progressively goes lower, and each step on the pH scale is a factor of 10 like 10 times hydrogen ions. So if I went from 2 to 1, that means I increased my hydrogen ion concentration by 10 times. So typically it's roughly 7.4 for the pH, and like it's good to know these numbers. I mean, relatively, like this is bigger on the outside, this is bigger on the inside, something like that. So this is what you call a sodium potassium pump, and it's one of the most important pumps in the cell. It's active in virtually everything, and what it does for you is it sets up a gradient. So it uses energy to pump sodium out to the outside of the cell. And so on the last slide we just said that sodium is already greater on the outside. So why would we do that? Well, we're pushing the sodium out and we're pulling potassium in because we want to set up a gradient where it's like so much bigger on the outside that it like has to come down naturally in, and it's gonna like flood inward. And I think so sometimes you'll set up the gradient, and because like that sodium is pumped out, glucose, for example, might want to piggyback and come back in, and that's called symport. So like the glucose can't get in until sodium comes in. Or there's an example of antiport where like calcium can't get out until glucose comes in, or something like that. So there's different ways that this gradient can work. And so the symport and antiport, that's an example of secondary active transport, because it's like we didn't directly use energy. We set up a gradient, and that gradient, we used the energy to set up the gradient, so that part was primary active transport. The energy actually used to set up that gradient. But then once we had the gradient, it was all just kind of naturally like flowing down afterwards. So that's the secondary active transport portion of it. Does that make sense? Okay. We have this thing called the Nernst equation, and that's to determine the actual potential inside the cell. So like what the kind of electric charge is inside. And so we might say, so most of the time potassium is the molecule or the ion that determines like the overall like charge or potential of the cell. And that's because like potassium has what's called leak channels that naturally allow it to flow into the cell. And so chemically we like to push the cells out by like a concentration gradient. It's bigger on the inside, so it naturally wants to go outward. But then electrically, because we're pushing out all this positive charge to the outside, remember positive and negative, they like attract. So because we pushed out so much positive, we create an effective negative on the inside, and that negative wants to pull the positive back in, and then you actually have this equilibrium. So equilibrium in biology is all about like the forward and the reverse rates are the same. So like this chemical force outward is the same as the electrical force inward, and that sets up this membrane potential. So this Nernst equation here is something that I would keep in mind or write on my cheat sheet or something like that, because you'll probably have an example of calculation on the test.
Okay, so the top one's the same as the bottom one, the equation, right? Just
so
like the bottom one's
right. So when you have this 1 over z, the z stands for the charge. So if I had chlorine, I would have a -1 because chlorine is -1 charge. Sodium is +1, calcium +2. So this is for like the potassium portion, because potassium is a +1 charge. So 1 over +1 is still 1. But I would know that this is the general form of the equation, and it gives you your answer in millivolts. So outside over inside. So 1 over z, 1 over the charge, times 61 times log of the outside over inside concentration.
So know the charges of the
Yeah.
the ions, and then we are given what the outside.
Yeah, he'll give you like numbers, and it's just pretty much like plug and chug.
Okay.
Yeah. Next, so the cell takes on what's called depolarization, and so that depolarization is necessary for all sorts of action to occur in the cell. So again, we have this sodium-potassium pump to set up that gradient, But then afterwards what we have is we set up like everything ready for the cell. Like there's all this sodium on the outside, there's all this potassium that's been pumped inward. And all of a sudden, like, we get some sort of signal that tells our cells, like, go. And so all of a sudden, all of the sodium will come rushing in. So these voltage-gated sodium channels open up, sodium floods into the cell, and then all of a sudden, because your cell was a negative potential before, like your resting membrane potential is negative 70 millivolts typically. So all of this sodium comes in, all this positive comes in, so negative 70 hits like a certain threshold, and it like rapidly shoots up to a positive potential. And so once it hits that positive potential, you're in what's called the absolute refractory period. So no other stimuli can like affect you now. You're like too far along the path to stop now. You're going to commit to whatever action. And so the sodium came in, and like gradually that potassium starts to balance outward. So the potassium will start to leave, and so depolarization, you kind of like went upward, and now you undergo repolarization. So your voltage-gated sodium channels, they kind of close off, and your potassium channels, they start to allow the positive Ks to flow outward, and then you start to get negative. Does that make sense?
So does the change in like when it starts decreasing, that's as soon as the potassium starts to go out?
Yeah, so it's built, so the depolarization is building upward
All the
because
way
we're
to the
getting,
top.
yeah, it's building up because all of the sodium is coming in, and then we start to open the potassium, but the sodium will close up before the potassium is done closing. So eventually, like, there's all this potassium flowing outward, and you get you become negative, and then you actually become a lot more negative, and that's called hyperpolarization. So hyperpolarization is a relative refractory period where I could stimulate like another action. It just needs to be a lot stronger than before. Yeah. So
which one is absolute?
absolute is like the first initial depolarization. So once I hit like, I want to say like negative 50 or something, once I hit that range, like I shoot up, and no matter what, I'm like gonna go all the way positive, no matter what. But if I'm like super negative because of the hyperpolarization, I could, in theory, if I had a strong enough stimuli, get back up to the original threshold. And so that's how action potentials work. And action potentials are present in like all of your motor cells and things like that. So anything that causes motion or needs to be doing any sort of action, as the name implies, like undergoes this process. This is the Krebs cycle. The Krebs cycle is important for making energy in the cells. It happens in the mitochondria. So remember when I said mitochondria makes ATP? So ATP is the functional unit that like creates everything. So like ATP was used to establish that initial gradient for the action potentials. ATP is used for like pretty much everything. And so the Krebs cycle, we use oxygen, so we generate a lot of ATP. It's like around roughly 30 ATP molecules per cycle. But then we also, anaerobically, we have this initial component called glycolysis, and that just uses glucose, but no oxygen. And so you make like a net of 2 ATP molecules. So you take 2 ATP and you make 4 in the first step, and then in the next step you like take all of the products that you made from here and you kind of do a little, processing, and you eventually make a lot more ATP. And so that's why it's important for us to have oxygen. This is cell cycle checkpoints. So your cells are constantly dividing when they're in the M phase. M is for mitosis, but then otherwise you're in interphase. Interphase comprises of G1, S, and G2, and so G1 is like growth, so we're making all of our organelles and whatever we need for like the next cell, And then we have S phase, which is replicating our DNA. And then we have G2, which is kind of like that second point where we check to make sure, one, we made all of the S phase, like we made all the DNA replicated, we have all of our organelles, we are absolutely ready, and then you move on to mitosis. But you don't need to know pretty much anything about these checkpoints. They're a little beyond the scope of this class. Types of tissue, yeah, 4 different major types that you guys will kind of run through in the future. I would just know the difference between like squamous cells are typically like, you know, the top of like different organs or something. They're like your, not to say like your standard epithelial cells, but they're more common. They're just kind of they can be layered if it's stratified squamous. But I usually just think of like skin and stuff like that. Cuboidal are a little bit tougher, and they can be in like glands or ducts and stuff. And then columnar are mostly for like secreting things. So columnar cells, they could be like in your intestinal tract if they're like secreting some sort of enzyme or some sort of, you know, mucus or something like that. For these terms, I would try to memorize them. They'll come up many times in the future, so I'm not going to run through them too much. Again, more terminology with like cancer-ish terms. Yeah, I just memorize these definitions as well.
Just the difference between dysplasia and neoplasia is neoplasia is a new abnormal disorganized growth, right? I mean, because he had said, I thought there was some confusion as to which one. I know neoplasia can be a wart, but dysplasia could also be a wart, correct?
Yeah, so like the neoplasia could be cancerous, whereas dysplasia is abnormal, but it's not cancerous. That's pretty much the bigger one. Metaplasia is like changing one to another, but that's pretty much the difference. So neoplasma, neoplasia, those kind of things are more cancerous. Yeah, think more cancer. This is just a slide of different cardiac muscle, and I think you'll go over it later. And again, this is an example of like the different types of cells, just to kind of orient yourself. Reversible implies that you can kind of somehow go to an adapted state that may or may not go back to normal, but typically it's just adapted and you kind of stay adapted. Or if you can't adapt, you just kind of die, like cell death, and just kind of goes away. This slide is very important. It's kind of a weird slide to understand at first, but I would just try to read through it a little bit more detailed. And if you have any questions after reading through it a little bit, we can talk more in the future. And this slide with all of these diagrams I would also run through. In general, like there's the difference between reversible and irreversible cell injuries or cell death, and when you have an irreversible cell death, you know, different things like spill out of your cells and different factors kind of accumulate. Whereas if it's reversible, all of these pathways just kind of help to, you know, reverse everything. So I would run through it, and maybe in a future lesson we'll kind of come back to it.
And the green boxes are clinical manifestations, but isn't it all clinical
they're like,
manifestations?
so like a clinical manifestation is more like a lab value chart, and then like everything else is more like this happens, but like, you know, yeah, it's more like it happens, but it's not necessarily like something that you would see like on a chart like straight out. Yeah. Again, reversible versus irreversible, different terminologies. So hypoxia and hypoxemia, they're slightly different. So hypoxia is a more general term. Hypoxemia specifically is about the blood oxygen content. Ischemia is lack or insufficient blood supply. Infarction, like think of like heart attack, so like the cell died because it didn't have oxygen. Reperfusion is a term for like if I cut off a blood supply and then all of a sudden I release it and like all that blood supply can come back, so it's returning, restoring. Necrosis is different than apoptosis. So apoptosis is like natural. It happens in all of our cells when things go wrong and they don't want to cause harm to other cells. So they just commit suicide, they die, they cycle away. Very, very clean. Necrosis is bad because like the cell will literally like explode and all of like the cell guts will just kind of spill out all over the cell, and it's very, very messy. It's very difficult to clean up from there, and like because once a cell like undergoes necrosis, there's a lot of cleanup to do afterwards. So you can.
Some examples of apoptosis would be like an autoimmune disorder.
So like it could be like a cell that may or may not have received radiation, let's say. So if I had one cell that underwent radiation right before it was about to go into mitosis, before it started dividing, in the G2 phase, we have DNA checkpoints all around the cell to say, okay, are you set to divide? Like, is there anything wrong with your DNA? And if it's like, oh, I think there's something wrong with you, it's like, just go ahead and, die. Like, go away. That kind of thing. Because if a cell has some sort of damage and it gets replicated in mitosis, that's what can lead to cancer. And there is a lecture on cancer coming up, so like that's a lot more exciting. A lot more details and like woo. So coagulative necrosis is just, as it sounds, coagulation, whereas liquefactive, you have an abscess forming. And I would know that these like different types, caseous has to do with tuberculosis. Fat deals with like fat saponification. And saponification is just kind of like a fancy word for like, it's almost like how you make soap. It turns fats into like different kind of components.
And liquefactive is, you can see it with the heart, correct?
So
or no, with the brain.
Yeah, you can see it with like a brain infarction or like kidneys, something like that. And then gangrene, this is with Clostridium perfringens. Also, you see it with like diabetics. And then lastly, telomeres are very important. They're on the ends of your DNA. And so every time you replicate your DNA, like your DNA kind of gets a little bit shorter and it cuts off the ends of telomeres. So telomeres start with like a set number of like replications that you can undergo, and it keeps cutting. And cancer, what's different with cancer is that cancer has the what's called telomerase, and it can actually restore those ends. So in a normal cell, if you get to the end of like your telomere, like the cell will die because it's like, okay, it's your time to go. But a cancerous cell will rebuild it, and you replicate more times than you were ever supposed to. And so they're what's called immortalized because they can just keep replicating like without ever dying or something like that.
Stem cells?
Cancerous cells. Cancerous cells. Yeah.
But can you define germ cells?
Germ cells are like the sex cells, so they're like
sperm cells.
Yeah,
Okay.
so sperm or eggs.
Yeah, we kept talking about sperm, but
Yeah. So genetics, is that okay to move on? Cool. Alright. So this is more in detail about like the molecular kind of everything stems from DNA. So with DNA, there's two anti-parallel strands. One runs, 5' to 3', and the other runs the opposite direction, 5' to 3'. And then you, with DNA, you have to, replicate it, or sometimes you have recombination when you're replicating, that kind of thing. But generally, it's very, very sacred. You don't want to tamper with it too much. And then you have RNA synthesis, so transcription, to become a single-stranded, let's say mRNA, messenger RNA, which will take a signal, like it'll take like a recipe out of like a cookbook, and it'll bring it down to the ribosomes where you have protein synthesis, and that's like translation. So proteins are, what make up like a lot of the different functions in our cell. They do almost everything. So again, like the DNA is like the cookbook, the master cookbook. The mRNA is kind of like a copy that I transcribed a little bit, and I just took it to like the kitchen to like make, but I didn't want to get the rest of the book dirty. And then lastly, like I make the protein, like I make a dish from the, from the recipe or whatever. So this is basically what I said before, but there's 4 base classical bases. So there's the T and the A, so thymine and adenine, they bind to each other. And then C and G, so cytosine and guanine, and so they always bind to each other. And so your A and G, those are called purines because of the structure. And then your cytosine and thymine are what you call pyrimidines. So pyrimidines have like pyrimidines have 1 like nitrogen ring, purines have 2. And always just know that like a pyrimidine and a purine always will bind to each other. So they're always in like equal numbers. So like A is always equal to the number of T, G is always equal to the number of C. Let's see, RNA. So protein synthesis, the way it works is basically a protein molecule. Once you have the mRNA from the DNA over here, it gets processed. So you get like a poly A tail and a methylguanosine cap on the other end. And so those are just things to kind of help direct it out of the nucleus, get it to where it needs to go, and make sure it doesn't get degraded by anything else in the cell. And so the mRNA gets a little bit processed, and we bring it over to a ribosome. And so your mRNA will have 3 bases at a time, and that's called a codon. And so your codon is like the code that you want to have, and then you have so it's kind of along the ribosome, and then you have the tRNA, which kind of looks like a clover shape, and it carries along with it a specific amino acid for that tRNA, and depending on what's called the anticodon, so like the 3 bases that's on the tRNA, it'll bind to the codon sequence of the on the mRNA, and then they attach, and then you keep moving along. So this is called translation. And so so again, just to repeat, like you have your mRNA loop, you've got your ribosome holding everything together. You read like three bases at a time, and those 3 bases are called a codon. You have a tRNA that's like your clover. It comes in, it has an anticodon that will bind to the codon, and it connects. And each tRNA has a specific anticodon, like specific code here, and it carries a specific amino acid with it. And so there's like many different combinations, but there's 20 amino acids, but there's like 64 different combinations, just to make sure you have everything. And then AUG, which codes for methionine, is always the first sequence that you start translating from, always. And then UAA, UGA, and UAG are the 3 stop codons. So it tells you when to like detach everything, and you don't need this kind of stuff anymore. Like you're done, the protein is finished, etc. Right. So that's basically what this is all saying.
Can I ask a really simple question?
No.
You're saying there's 64 possible codons?
Yes.
And you said it was 4 times 4 times 4. Where does the 4 come from?
So there's 4 possible bases, A, C.
Oh, that's what it is.
Yeah,
Thank
so there's
you.
the 4 different bases. Remember in RNA you have uracil, a U instead of a T. And the reason for that is a thymine has an extra methyl group sticking out from it, and cytosines tend to like undergo like a process that makes them look like a uracil. And so it doesn't happen in DNA, but it can happen, like they can get mutated or switch to a uracil easily, but it's an RNA, so it's like not as bad if it like changes. But if it happened in DNA, that'd be really bad. So there's the 4 bases, and then you had to come up with like 4 times 4 didn't make sense to make the correct number of like amino acids. 4 times 4 is 16, so you had to have like another 4 like combination to be able to encompass all 20 amino acids. It's kind of a weird way to think of it, but yeah. Okay, so this is an example of what's called a mutation, and so there's different kinds of mutations in the DNA, RNA, and so typically your DNA, if it gets mutated, it'll correct itself somehow. It'll repair itself. So hardly ever any mutations in the DNA relative to like RNA and other things like that. But if a mutation does happen, it could change a single base. It could insert something, and that's what's called a frame shift mutation, because if you insert one, everything gets read like at a different interval of 3. Or you could delete one, and then, you know, again, that's another chain. So a single base pair mutation, that's called a SNP, a single nucleotide polymorphism. Polymorphism meaning change. This chart, and the ones that'll come later, I think are just pretty easy to like throw on your cheat sheet, but not this one specifically. It just outlines what we're going to talk about later. In general, so you have single gene disorder, so like you have 2 genes, or 2 copies of each gene, mom and dad, and a dominant gene would be like 1 copy is enough to make you go bad. Recessive, 2 copy, like 1 is not enough, 2 has to be bad. X-linked is like the one from, you know, the X chromosome will, you know, if it's recessive, it needs like 2 X's. If it's dominant, just 1 X. Translocations, deletions we'll go over. And then aneuploidy is a different number of chromosomes. Not like a complete set of chromosomes. Like, it's just like plus, you know, plus another chromosome 13, plus another 18, plus another 21. But it's not like plus 23 other chromosomes. That's called polyploidy. So polyploidy, like, it's not viable. But the only aneuploidies that are, so trisomies are 1 extra, so 13, 18, 21, those are the 3, like, somatic ones, so non-sex cells. And then you could have 47 XXY, which is a trisomy, and that's Klinefelter syndrome. And then there's Turner syndrome, which is the only, like, monosomy that's viable, and it's 45X. And we'll go over those in a second. This terminology, have you guys, are you kind of familiar with it? So a locus is like where it is on the genome, okay, a location. Allele is 1 copy of the gene. So like mom's allele, dad's allele, like that make up, you know. A genotype is like the genetic code that tells you, like, you know, I'm like capital TT, if you guys have seen like a pedigree or something. But then a phenotype would be like, because I have 2 capital TTs, I'm tall, or something like that. Penetrance is like, if you have this genotype, what is the chance that you actually have, like, even though you have capital TT, what are the chances you actually become tall? Like, for example, a haplotype is like the different alleles on a single chromosome. It's like you have multiple alleles that kind of make up a set of your chromosome. So it could be like, you know, what does the actual chromosome look like, sort of. Recombination and crossover, that's like how you get new genetic information. And a SNP, SNP, single nucleotide polymorphism, that's changing 1 base.
Going back to penetrance, like,
Yeah.
how do you take into account the environment for the phenotype not to follow the genotype?
Sometimes. So like there are some like genes that 100%, no matter what the environment, will have the disease, like Huntington's disease. That 100%, if you have it, you will manifest it. but other things like if you had like a syndrome that's mostly active in like guys, but you're a girl or something, like you might not actually manifest it. So it's not always 100% with genetics alone. Like there are environmental factors that kind of mediate the effective penetrance of things. So structure of chromosomes, you have a P arm, and that's the short for petite, and Q because it was the next letter in the alphabet. So long arm. And so you've got a chromatid is like one copy of the chromosome, sort of. Like, I usually think of it as like, like most of the time in your cells, your chromosomes aren't actually like X's. They're all kind of scrambled around. And they're only X's once you get to mitosis, once you're dividing. And then you have a centromere that's like in the middle that connects everything together. But typically a chromosome is more like, it's just like a little that. And then you like replicated both ends because later you're going to pull it apart in mitosis. Does that make sense? Next you have stages of meiosis. So your like spermatogonium or your oogonium, like the original like egg or sperm like first cell, those cells can do mitosis to make multiple, but then from there they undergo meiosis to meiosis 1 and 2 to make either 4 sperm per one like spermatogonium, or it's 1 egg per spermatogonium. And so the difference in male and females is that males, like, you start making the sperm once you hit puberty, but then in females, you have all of the eggs that, like, you will produce. They're ready, but they're all just frozen up until puberty, and then you start ovulation, and that's when you kind of release it to go through the rest of the cycle. So women have a process called atresia, where like they lose eggs each month, and then 1 egg gets selected, and so most of your eggs are frozen in meiosis 1. Once it gets ovulated, it can go up to like metaphase 2-ish, and then once it gets fertilized, so like once that sperm hits, then you can actually be, hit and become an actual egg. So it's a little bit different than how elementary kind of biology is taught, but then like in actuality it's a lot more complicated in females than in males. And the reason why it's 1 egg per instead of 4 eggs is because like the other 3 things actually it's usually 2, they're polar bodies, so they're kind of like leftovers, and they gave everything else to the egg to make it bigger. So the egg is the biggest cell in the body. It's like 140 micrometers or something, and most others are like 10-ish. Non-disjunction can occur if you have the cells like pair up in different ways. So like if you have they can instead of separating like in meiosis 1 like they should have, it'll both go into 1. Or in meiosis 2, instead of separating, it'll like both go into 1. And so like there's different combinations of how non-disjunction can work. So this is Down syndrome. So trisomy 21, one of the viable ones. Turner syndrome, is the only viable monosomy. Klinefelter. So these disorders, like when they don't really exhibit the correct like secondary sex characteristics, and that's just like development of whatever they would have developed if they were normal. But typically like intelligence is roughly the same, but they are probably sterile because of like the abnormal chromosome number.
We need to know what the 45 and 47, what does that stand for?
So number of chromosomes. So typically a normal person has 46, 23 by 23, but then like you can say, like 45 XXY meaning, or 47 XXY, so you have an extra X, or 45X, you lost the 46th sex chromosome. Crossing over, what happens is in meiosis, your cells, in prophase 1, they all line up with their homologous pair, So remember in S phase, we duplicated our DNA. So I have like, let's say this is like an X, and then I made two X's. So these two X's, they, like one from